talk a little bit about resting state fMRI, but my focus will be on connectivity in autism spectrum disorders. Now, autism is in the media quite a lot, so you probably know that it's a disorder with increasing prevalence, very high prevalence. Actually, 1 in 88 kids is the most recent estimate, and it's diagnosed on the basis of behavioral criteria, although we think it's a neurological disorder. That's something I'll come back to towards the end. Just very briefly, what are the current diagnostic criteria? They're actually going to change a little bit with the DSM-5, but autism is primarily diagnosed based on socio-communicative impairments, including restricted and repetitive behaviors. So the picture on the right shows that it's not always good to have all your ducks in a row. Now, it's a socio-communicative disorder, but when we look at the literature on neuropsych profiles and autism on the whole, it's actually much broader. So there's evidence for a very broad and atypical profile of neuropsychological, cognitive, and sensory-motor impairments with some island strength. And there's a lot of variability and a lot of heterogeneity within the disorder. In fact, it's generally accepted that there are multiple etiological pathways that lead to a phenotype that we call autistic. So people started talking about the autisms. That is a catch-on. We talk about autism spectrum disorder these days, and that's actually the term the DSM-5 will use. So it's a set of disorders. We know that genes play a very heavy role in the causation of ASD, but there are in fact hundreds of genes that have been identified as playing some role, being associated with ASD, so the genetic literature is by far not conclusive. What's interesting is that many of these genes have something to do with synaptogenesis and circuit formation. And that brings me to the topic of my talk, which is connectivity. Now, why would we study connectivity in autism? Why don't we just apply a more standard adult neuropsych approach to autism? Well, we can, and people have tried it. There are attempts to kind of localize autism and look at it with the standard neuropsychological view, like hole in the head approach that we know from adult. Sorry, apologize to all neuropsychologists who are sitting here. That's obviously a big simplification. But there have been approaches, for example, linking ASD to specific localized impairment in the amygdala. The cerebellum was a topic some 1 to 2 decades ago. However, when we look at the broader literature, just looking at the anatomical MRI literature on ASD, what we see is that pretty much anywhere in the brain have there been findings on some kind of abnormality. You don't need to try to read all this. I gave up completing this slide. The font would have gotten too small. There are lots of more findings, but I think you get the general idea. There's pretty much, there seems to be something wrong anatomically, pretty much anywhere in the brain. So within the past at least half decade or so, there's been growing consensus that if we want to understand ASD, rather than trying to look for it in A, B, or C, one particular part of the brain or a small set of brain regions, we have to look at how different brain regions communicate, talk to each other. In other words, the connectivity, network connectivity. And that's what I want to talk about now. Now, this is supposed to be a symposium on fMRI, but I have to talk very briefly about the anatomical side, about diffusion tensor imaging, because obviously connectivity is not just functional, it relies on anatomical tracts, and diffusion tensor imaging is the technique of choice to look at the anatomy of white matter tracts. So, in very, very simple terms, how does DTI work? Well, we look at water diffusion, and if we have diffusion that is unhampered or restricted by tissue organization, it's isotropic, as indicated by the circle. It's the same in all directions. Now, if we look at the nicely organized fiber tract, it's different. It's anisotropic because it is restricted by the fiber organization, by the membranes and the myelin sheaths, primarily, that we find in axal tracts. There are other DTI indices that are also affected by tract organization. Mean and radial diffusion should be low. That's good. You want them to be low because, for obvious reasons, overall diffusion is reduced, and the radial diffusion, orthogonal to the main axis of diffusion, is also reduced. So these are the three indices that I very briefly want to talk about. This is a study from my lab looking at these indices in children and adolescents with Ph.D. Uses tract-based spatial statistics. This is really a technique that makes it easier to do group comparisons. Pretty smart way to normalize the data. The green stuff that you see there, that's a tract skeleton. So that shows the major tracts in the brain, and then you could do group statistics just for the skeleton. And the red spots that you see, these are areas of the skeleton in tracts, different tracts, where the kids with ASD show reduced fractional anisotropy. And you can see it's not just one tract in it, but it's in both hemispheres, and it's quite distributed, affecting multiple tracts. When we look at other indices of white matter compromise, increased mean diffusion, you again see this picture of highly distributed and very widespread effects of white matter compromise, and similar when we look at radial diffusion. One thing that I want to add here very briefly is that in the typically developing brain, we indeed see FA going up way into adolescence. And here we're not just looking at prefrontal white matter, but we're looking at all of these tracts. We looked at them separately. These effects are actually quite robust still, going into the late teens. And then mean diffusion goes down significantly in TD kids and adolescents. Compared to that, these age-related effects seem to be somewhat diminished in ASD. And that's potentially worrisome, but I don't have the time to go into this here now. So, there is consistent and widespread evidence of white matter compromise in kids and adolescents with ASD. I showed you only one study, but there are easily 20, 25 studies now that are quite consistent. And that's true not only in cortical tracts, but also in deeper tracts that support sensory motor functions. Just one example here, that's a study looking at the corticospinal tract. These are axons going from primary motor cortex into the spinal cord. We only follow them here into the cerebral peduncle. And in these tracts that support motor execution, nothing cognitive, nothing sociocommunicative, we also see evidence of white matter compromise. Significantly increased mean diffusion. Sorry about the boom because of my jacket. And significantly increased radial diffusion. All right, now let's talk about functional connectivity. fMRI. The picture actually is an example of functional connectivity of the subway system in my hometown, Hamburg. Much more functional than the trolley system in San Diego. So, very simple principles of fCMRI, as we call it, functional connectivity MRI. We really look at bold signal correlations across different parts of the brain. So this was the very first fCMRI study from my lab, and we looked at a connectivity of primary visual cortex in red up there. And you can see a time series extracted from this seed, and then you can compare it to the time series in black in some other part of the brain. You can see inferior frontal cortex, and that's from a TD participant, and that's from a participant with ASD. And then you can make whole brain maps of the correlation within each group, and also the difference in correlations between groups. And what we were excited back then was to find that bilaterally inferior frontal cortex reduced correlations, reduced functional connectivity. We were excited because that was about the time when people started talking about the mirror neuron system being impaired in ASD, and that seemed to fit, although of course this is not, we're not looking at individual neurons here. We don't really know whether this is the neuron. Now, around the time when we were working on this study, a milestone paper came out looking at functional connectivity in ASD by Marcel Just. That was connectivity during sentence comprehension, and what they found was reduced connectivity between a bunch of ROIs that are shown here. No, okay. Sorry about that. The middle panel, and the conclusion was that there was something like general underconnectivity in the autistic brain, and that was followed up by a bunch of studies, mostly from the same group, that replicated this finding, looking at different functional domains using different types of tasks, all listed there, and also studies from other groups that supported underconnectivity in ASD. So that's wonderful. That would be one of the rare examples where the field actually agrees and has consistent findings in ASD research. First firm finding, underconnectivity. Now, the paper actually came with a question mark. And I have to disappoint you now because I want to show you a lot of findings that are inconsistent with this. There's a growing number of studies that don't show this functional connectivity, and admittedly, quite a lot of these are from my lab. I want to just show you a few of them, and then I want to explain to you why I think this is so. So this is an example of a study where we had an activation paradigm, semantic decision task, that activated Broca's area in both ASD and TD groups, and we used the area of shared activation for this task as a seed to do functional connectivity analyses. That's the black cluster circle there. So the little black cluster inside the big circle, that's the seed. And you can see that in the TD group, there's a relatively distinct connectivity pattern. You always see homotopic connectivity on the other side of the brain. In right IFG, that is expected from the TD literature. And then we also see the connectivity with posterior perisylvian cortex Wernicke's area. Now, above the ASD group, you can already see that these connectivity patterns appear much less distinct. And when we do a direct group comparison, we see actually a lot of overconnectivity, increased both signal correlation in the ASD group, especially in the temporal lobe. Let me give you one more example. Obviously, that's very consistent with the results I showed you before. That's an example looking at an imitation circuit. And this study actually used 3 seeds taken from the literature, the sites that are most likely considered to be important for imitation. We initially actually called the study mirror neuron system, and then we got clobbered by reviewers, so we kind of pulled back and called it an imitation circuit. So the seeds are highlighted here in inferior frontal cortex, inferior parietal lobule, and superior temporal sulcus. And you see color-coded the connectivity patterns of the two groups. And there's quite a lot of convergent connectivity in the TD group. The black clusters, that's where, in a sense, all the 3 seeds come together. They have shared connectivity. You also see that in the ASD group, but the more striking finding on the between group comparison was that the ASD group showed atypical connectivity with dorsal frontal cortex, over-connectivity with dorsal frontal cortex. And we did follow-up structural equation modeling, which is supposed to be tell you something about the causality of the interaction between the regions. I don't know whether I actually buy the causality part, but it definitely shows us that this atypically activated dorsal prefrontal cortical region had some modulating impact on the imitation circuit in the ASD group that it didn't have in the TD group. Might be compensatory, we could not actually look at that in this particular study. We have the sample size, and also we didn't have the behavioral data to support that. Now, in a follow-up study, Patty Shee, who did this particular study here, looked more closely at one of these regions of the imitation circuit, the posterior superior temporal sulcus. That's an area that's very interested. It's not just important for imitation, but it's an area that's important for biological motion perception, language in the left hemisphere, and so on and so forth. And what she did was she identified 3 subregions within posterior superior temporal sulcus that were actually maximally distinct in terms of their time series. And they're color-coded here. So we have a rostral region, we have a middle region, and a caudal region. And then the connectivity patterns of these are shown using the same color code. And you can see that there's a great distinction between the middle seed, or subregion, and the rostral and the caudal ones, which actually have, rostral and caudal ones are quite similar. You see quite a lot of overlap between the bluish clusters, but hardly any overlap with the red cluster, the middle seed. So there's differentiation here, and that's in the TD group. These are children and adolescents. These are the, these are the results in the ASD group, and you can see there's a lot of black stuff there, or very dark stuff. That is atypical overlap, shared connectivity, reduced differentiation in the ASD group. Now this reduced differentiation was associated with overconnectivity in the ASD group for all 3 of these little seeds. Again, using the same color code, you can see the extensive overconnectivity in the ASD group. Looking at this differentiation, we use the differentiation index, which is in a sense inverse of the correlation coefficient. And we see that in the TD group, differentiation goes up with age in childhood and adolescence. So if we ask how different are the both time series between these different seeds, goes up with age significantly. Or we ask how different are the connectivity patterns between the 3 subregions, goes up significantly with age in the TD group. In the ASD group, these effects again are diminished. So differentiation is reduced, and the age-related changes seem to be also diminished in ASD. Now you could ask, is this something we only find in STS? We actually have a study looking at Broca's area, looking at different subregions, the differentiation within Broca's area. And without going into the detail right now, we have a pretty consistent findings that the differentiation between the subregions is reduced in autism, and there's also widespread overconnectivity of left inferior frontal gyrus, and in fact also right inferior frontal gyrus in ASD. So we could conclude that the diffuse overconnectivity that we find has something to do with reduced specialization or differentiation of cortex in ASD. Now, however, I've kind of lured you into this kind of conclusion, and I haven't really explained to you why we have this rather hair-raising inconsistency with large parts of the fMRI literature that I presented first. So I don't even need to explain this. Tom already talked about Bart Biswold's study. The first resting state functional connectivity study. So you can map out the motor system, for example, using resting state data. And follow-up studies have shown that the best frequencies to do that, when we talk about fMRI data, are between 0, well, I would say 0.001 and 0.1 hertz, very low frequencies. That's one study showing that these low frequencies are specifically reflective of network connectivity, whereas slightly higher frequencies are noisier. Probably has something to do with slow fluctuations in local field potentials. And these fluctuations have been shown to be affected by learning and have been related to Hebbian plasticity. But again, the important thing is that these correlated fluctuations occur spontaneously in the absence of a task. Now, when we go back to the ASD literature, the studies that I talked about at the beginning, the underconnectivity studies, are pretty much all of a very different type, because they look at the correlation of task-driven effects. Whereas the studies that I showed you after that are kind of more conventional fMRI studies looking at intrinsic low-frequency fluctuations. And these tend to actually yield over-connectivity effects in ASD. And we looked at the literature to see whether that interpretation is actually true. So in this meta-analysis, we found, grouping studies as underconnectivity or overconnectivity studies, color-coded differently, that the methodological fingerprint is actually very distinct. In particular, underconnectivity studies don't tend to perform low-pass filtering. They don't remove task effects from data, and they usually only look at some regions of interest rather than looking at the whole brain. So they usually only look at network nodes. And currently, we're following this up by analyzing some of our own data sets to test whether these methodological variables actually have the effect on the results that you would expect. And don't even bother about this table. Let me just summarize this. The main findings. So when we look at higher frequency task-driven correlations, we tend to find underconnectivity in autism. When we look at low frequency intrinsic correlations, we tend to see overconnectivity in ASD. And these overconnectivity effects tend to occur outside areas that are activated for the particular network that we're looking at. So, for example, the outside ROIs for the imitation network that I talked about earlier. Now, this is all methods, and you're probably already drifting off. Tell me something about autism. So what does that tell us about ASD? Well, here's an interpretation. What we probably see in ASD is a dual impairment in the sense that when we look at the connectivity within a hypothetical network, AEC, it is reduced. The connections, at least functionally, DTI, we don't have that evidence yet, functionally, they are reduced within the network, between the network nodes. However, there's a lot of potentially residual connectivity, diffuse over-connectivity or connectivity with other parts of the brain that don't play a major role in the typically developing brain in that particular functional domain. Oh, you're scared. That was great. And I gave you some examples for that in earlier studies. So what we could say is that we have impaired network sculpting in ASD. And that relates to Mark Johnson's approach to network formation, the interaction of progressive and regressive events that I would hypothesize are effective in ASD, which then leads to inefficient network organization and noisy information processing. Now, that's a reasonably simple model. I have to admit that some of the data that we have in my lab are actually not always completely consistent with that. This is one example of a study looking at thalamocortical connectivity oscillation based on DTI comes out pretty nice, as in previous studies on adults. DTI shows underconnectivity for different cortical seeds, consistent underconnectivity. fMRI also shows mostly underconnectivity between cerebrocortical regions and thalamus, except for right temporal lobe. And don't ask me what that means, we're trying to figure that out right now. But this is not consistent with the idea of diffuse overconnectivity. So not everything is consistent with this simple model. It's probably more complicated. All right, now this is resting state data. And there, I could go some other route now if I had more than the 90 seconds that she's giving me. Bash the resting state, so to speak. We don't really know what happens in the brains of kids with ASD when they are in the resting state, and there are potentially severe confounds, but I will not go there right now and try to jump things with it. And goes, oh, now the red card. Oh. All right, I just want to say a few things of where we can go with future applications of fMRI. I talked about multimodal approaches already. Surprisingly, there's not a single study in the ASD literature that has combined DTI and fMRI today. Not a single published study. The one that I just showed you is not published yet. That's very surprising and will probably change soon. So that's definitely something important to tackle. Data-driven approaches. So this is an example of a study using independent component analysis, actually using the approach that Tom, Steve Smith's approach that Tom already showed you. And what we found here was that there are differences in asymmetry in these data-driven functional networks that we detect between ASD and TD kids. So we find 17 non-noise components in resting state data, and 10 of them show group differences in asymmetry. Now, one would think probably 5 and 5. 5 are more leftward, 5 are more rightward. But that's not the case. All of them, exclusively, all 10 of them show a rightward shift in asymmetry in ASD, which has made me a lot more interested in asymmetry in ASD than I was maybe a couple of years ago. There seems to be actually a pervasive abnormality of hemispheric asymmetry in ASD. We're also looking at local connectivity. There have been some speculations that local connectivity is actually enhanced in ASD. We have some early findings that suggest that it might be true, but I'm not quite sure. That's the end of the story, but what the data-driven approaches allow us is to find things that we wouldn't expect. There are correlations with diagnostic severity, but there's an atypical age-related change. Local connectivity goes down with age in TD kids, but in fact it goes slightly up in ASD with age in kids and adolescents. Okay, let me skip this here. Some more approaches that we are working on right now are increased spatial resolution, both looking at cortical organization with GABA, MR spectroscopy, and using enhanced diffusion techniques to look at cortical organization. Why? Why is all this interesting? Now, one thing that the field needs is biomarkers. I mentioned that diagnostics is still done in terms of behavioral criteria. That's a big problem. So we need biomarkers, and connectivity approaches are really one of the best places to look for biomarkers. And the biomarkers we need for diagnostic classification, ASD versus TD, but more importantly, to find subtypes of the disorder, which currently we don't know. And the perspective is really that these subtypes will actually be linked to genetic and or environmental causation in more straightforward ways than the ASD population as a whole, which of course will then be the first step towards targeted treatment. All right. Thanks to you for listening, and thanks to all these people who helped and the funding.